No, it's always really good to hear Aldo. Hi, Jamie.
Okay, I think it's a minute past 1, so let's make a start. Welcome everybody. It's great to welcome an alumnus speaker. Mateo did his PhD here a few years ago. I think he finished about 3 years ago and did his PhD with Dora, and now he's over in the US in New Jersey at Rutgers, and he's going to be telling us about the work that he's been doing. So over to you, Mateo.
Thank you, Chris. Yeah, so actually yesterday night I was going through the slides one last time, and I was just really sad that I cannot be there. You know, I really wish we could. I will just have to come back in a couple of years or something. Yeah, lots of memories. Like Chris was actually the first person I had a beer with at Sussex on induction day. So yeah, it's a lot of memories being here. Okay, so yeah, today I'm going to be talking about the research I've been doing at Rutgers at the Center of Alcohol Studies since I moved here after my PhD. Going to be talking about interoception and how we are studying the interoceptive mechanisms of alcohol-related behaviors and other interesting stuff that we can maybe use clinically in the future. So I think that we can skip this whole bit because everybody at Sussex knows more or less what interoception is. I will just say that I'm studying interoception from different points of view. So usually, especially the work that Sarah Garfinkel has been doing focuses on high order levels of interoception, so interoceptive sensibility or the perception of heartbeats. But we are also studying it through different orders. I also study how basic cardiovascular signals, for example, measures of heart rate variability, support cognitive processes because it just allows us to study body-brain connectivity in a more physiological way. And we also study how discrete signals, how baroreceptor firing and so on, supports cognitive processes. And we do that by pairing stimulus presentation with cardiac cycle to study how the heart supports different emotional and cognitive processes. So now that we have that out of the way, maybe we can just get started with the whole point of interoception and addiction. There's been a lot of papers. It all sort of, I mean, in the animal literature, it's been going on for many years, even before we thought about it from a human perspective, the whole point of interoception. But yeah, it all sort of started in 2006 when they found these insula damages provoking smoking cessation in smokers, in humans. And since then, there's been a lot of neuroimaging studies linking brain areas related to interoception and processing of bodily sensations and reward processing and decision making. However, there is not really a lot of research. I mean, that has generated a lot of interesting hypotheses. Already, Gray and Critchley pointed that out in 2007, that the interoceptive representation of alcohol stimuli could drive alcohol-related behavior, shaping craving and consumption. But really, we are missing a lot of research across dimensions of interoception in relation with more clinical populations or addictive processes, basic behavioral correlates of addictions and substance use, and also pharmacological studies. There is very few studies examining the interoceptive correlates of pharmacological administration in humans. So that is sort of what I'm trying to do now. I'm just trying to understand if physiological interoceptive signals can shape alcohol-related behaviors. So we sort of started this line of work during my PhD when we did one of the first alcohol administration experiments related to interoceptive awareness. We gave participants either a placebo or 2 different doses of alcohol. We measured their feelings after alcohol consumption and then measured interoceptive awareness using the heartbeat discrimination task, where people hear 10 beats that can be synchronized or non-synchronized with their own heartbeat. And out of that, we just get an index of how good they are at feeling their own heartbeat or at synchronizing or at indicating whether their heartbeat is synchronized with an exteroceptive beat. And what we found was quite interesting. We found that when we gave alcohol to people, the more they felt, or the higher their metacognitive accuracy on the heartbeat discrimination task, the more they felt lightheaded. And lightheadedness is something that is actually quite interesting as an index, because it's been shown, Dora did that in the early 2000s or late 90s, that lightheadedness, just the feeling of lightheadedness after being administered alcohol, is what helps people discriminate whether they have been administered a drink, or an alcoholic drink, or a placebo. So it was interesting to see that depending on how good people are at feeling their own hearts, their own bodies, or how aware they are of their bodies, they are also better able to perceive these acute alcohol effects. So that was pretty interesting. When I went into Rutgers, the first thing I tried to do was to replicate this with a little bit more of a complex design using a double session, and also including a lot of other measures. So we measured anticipated alcohol effects, so how people expect to feel after drinking, and we measured that at the beginning of the experiment, and then changes in mood, changes in craving, this change in lightheadedness, and we had people complete a baseline measure of interoception, and then 40 minutes after alcohol administration, so we get an index of change in interoception due to alcohol administration. We found, I mean, I don't take this result, I really take it with a grain of salt, but we found that alcohol increases the ability to perceive heartbeats. It increased interoceptive accuracy, even when controlling for changes in heart rate. But the most interesting bits come when we try to replicate the previous findings. We found that the more interoceptive awareness, so metacognitive interoceptive awareness and confidence increase after alcohol administration, the more people felt these lightheadedness effects on after being administered alcohol. And actually, we had a sensitive null on the placebo condition. So it seems that these effects are really driven by alcohol administration. We also found that the more interoceptive accuracy and metacognitive interoception increased, people reported more positive mood after alcohol administration. So it also seems that increasing body-brain connectivity due to alcohol just changes the way you feel after drinking. And we also found this super interesting result, that changes in interoception after drinking negatively correlate with negative alcohol expectancy. So basically, those who felt a greater disconnect in body-brain connectivity, or who were worse at feeling their own heartbeat after being administered alcohol, had worse prior expectancies when they came to the lab about how one feels after drinking 3 beers or whatever. Whereas those who, after being given alcohol, felt better their heartbeat, they had less negative expectancies. They didn't expect to feel bad after drinking. And that's something we are trying to put in context here, and we are trying to build a model about how alcohol affects interoceptive processes to shape perceived alcohol effects, to shape positive mood, and on the other hand, shape negative alcohol expectancies. And what we think may be happening here, if we go back to a very basic animal perspective of interoceptive reinforcement learning, is that those interoceptive signals are just working as an unconditioned stimulus. You have to think that alcohol expectancies are usually defined by your experiences, your prior experiences when drinking alcohol or when seeing someone drunk. If you drink alcohol and you throw up, you're going to have negative expectancies, right? So what we think is that depending on the interoceptive experience that you have, how your body is feeling after drinking alcohol, that is also feeding into the model you are building on how you are going to feel in the future when you drink alcohol. So these interoceptive experiences will just be shaping expectancies and future alcohol use. That is actually right now, we're actually trying to replicate it with a different interoceptive task. So we will see if we manage to replicate this effect. Then moving on, again, I was really interested in going to the very basics of, I call it European style alcohol cognitive biases, when all the work that Matt Field, that Dora, that Reinhard Wiers have done on alcohol pure reactivity, and seeing if we can adapt that to study how interoceptive processes support alcohol-related behaviors. So on top of looking at alcohol cue reactivity, for example, with attentional biases, we can also look at physiological changes. And this is something that our lab has been doing for quite some time. And what we do is we look at changes in heart rate variability, which is the variation in time between different heartbeats. And those variations in time can be decomposed in different frequency bands. And in our laboratory, we are particularly interested in this 0.1 Hertz heart rate variability measure, which is just how much of the variability in interbeat intervals, in RR intervals in your heart, occurs at this very specific frequency band. And why is this frequency band so important? Because it's a proxy for baroreflex sensitivity. It's a proxy for how much communication there is from your heart to the brain and back. So we consider this 0.1 Hertz heart rate variability as a low order interoceptive signal. And as it turns, in the past, my PIs have found that this 0.1 Hertz heart rate variability measure changes after alcohol administration. They've also found changes in 0.1 Hertz after or during presentation of alcohol stimuli. So it seems that it is possible to study physiological reactivity as a measure of alcohol cue physiological bias, really. And my idea with this set of experiments is to understand if physiological responses map onto alcohol cognitive biases. There is also this concept of alliesthesia that I'm very interested in. The idea that the pleasantness of a reward depends on the state of the body. Easiest way to understand it is with hunger. When you are hungry, food tastes better. If you eat a lot, then food tastes worse. And the most interesting thing is that we can actually modulate those bodily states to change the value of rewards. And that is something that a lot of people have done. For example, Dora has done that. Our lab has done that. And it's what we call alcohol priming effects. When you bring someone to the lab, you give them a small dose and of alcohol, and then you just study how they pay attention to alcohol cues. But it's just people don't call it alliesthesia. I do think that is an alliesthetic process. And already Martin Paulus, quite some time ago, it posited that interoceptive processes will be crucial in understanding how the changes in reward values operate through interoceptive processes to shape addictive behaviors. And again, the easiest way to understand it is when you are not hungry. We call this the peanut cocktail effect. You are not hungry. You have a peanut, or you have some chips, and then you're hungrier than you were before because you have primed your body. So that is sort of what I was trying to chase with the following experiment. We just published this earlier this year in Psychopharmacology, and again, just trying to see if the effect of alcohol on bodily states, on changes in heart rate variability, could support alcohol attentional biases. So what we did is we brought participants to the lab in 2 sessions, and we measured their physiology at baseline, then gave them alcohol, and then measured their physiology again. And we are trying to see changes in after alcohol or placebo administration in the 0.1 Hertz heart rate variability. And after that, we had a measure of cognitive biases using a modified flanker task that Kiki developed, where we just study alcohol attentional biases. And what we found was really interesting. We found that there is some quite large inter-individual variability in 0.1 Hertz heart rate variability changes after alcohol administration. Some people show an increase, although most people tend to show a decrease in 0.1 Hertz heart rate variability. And interestingly, depending on how much this physiological index changes, participants showed more or less alcohol cue reactivity. So what this is telling us is that depending on how your body is reacting to alcohol administration, it's shaping the values of rewards, and we can observe that through changes in attentional biases towards alcohol cues. And again, I think I had a sensitive null on the placebo condition for this. So keeping that in mind, that we were able to show that depending on how your body responds to alcohol administration, the value of rewards changes. We were also interested in in adding a risk factor to this idea. And in our lab many years ago, like 15 years ago, they ran this super big project where they brought people to the lab who had a family history of alcohol use disorder. They brought people in who have at least a first order relative, like father, mother, uncle, who has a history of alcohol use disorder. And we know that these people have like 9 times higher risk of developing alcohol use disorder in the future. But turns out they also show, just because of their family history, they show increased alcohol attentional biases. They show also increased physiological changes in 0.1 Hertz heart rate variability after alcohol administration. And turns out they also show increased activation in the insular cortex in the presence of alcohol cues. So what we wanted to see is if we can observe or we can understand a physiological basis or interoceptive basis for this increased risk for alcohol use disorder. So we published this also like last year. Again, this is not data I collected. This is a big R01 grant that Marsha Bates got. What they did is they brought into the lab 158 people for 3 sessions, and out of those 158, 106 of them had a family history positive. I still don't understand how they managed to do all this work. It's insane. And basically, again, they did this thing where they measured physiology at baseline, then administer alcohol, placebo, or control, and then they presented people with pictures of alcohol positive, negative, or neutral stimuli in a blocked fashion, and obtained these ECG measures. And the idea is we want to see changes in physiology from baseline to each of the image blocks as a function of alcohol administered. And then after a break, participants had to recall as many pictures as they could from each of the categories. So what we were trying to do with this analysis is understand if changes in physiology at 0.1 Hertz can shape memory biases, so how many pictures of each category people remember, as a function of stimulus type, beverage, and family history. So we threw everything on a very big linear mix model, decomposed it through multiple steps, and we ended up finding one and only one positive correlation in people who were administered alcohol and who have a family history positive. We found that the more, or depending on the change in 0.1 Hertz heart rate variability towards alcohol cues, they were able to remember more pictures. So what this is telling us is that for this particular sample of people who are at higher risk, if you give them alcohol, those who show higher changes in cardiovascular afferent signaling in the presence of alcohol cues are more likely to remember them. So again, we are showing the coupling of physiology and attentional biases in this very particular sample. And we take this as an index of risk for future alcohol use. And interestingly, we also had measures of how many drinks people, no, how subjectively intoxicated people felt at the end of the experiment. So we threw that into the model, and what we found was very interesting. We saw that in this sample of family history positive, when we administered them alcohol, those who felt not too drunk, like were on the left side of the spectrum and didn't feel too drunk after alcohol administration, they do show this positive relationship between physiological responses towards alcohol cues and memory. Whereas progressively, as people felt more drunk, the relationship between physiology and cognition disappears. And that, again, I think brings back to the whole idea of alesthesia. And depending on how people feel their bodies after alcohol consumption is going to shape the value of rewards. When you are not feeling too drunk, you feel like having some more, the value of the reward is high, and that is reflective on the relationship between physiology and cognition. Whereas as people feel more drunk, the relationship between physiology and cognition disappears because probably the value of the reward disappears. So that's interesting. We can start building the model. So we can see that we have these high order measures of interoceptive awareness correlating with more subjective indices of alcohol responses. whereas this more basic physiological signal can shape reward values, observable through attentional biases and memories, and these bodily perceptions of feelings of intoxication can also weigh into the relationship. So before I move on, do we have any questions about this? Quickly. Okay. Next. So I'm changing a bit topics right now, but I will get back to alcohol in a second. So again, as I said at the beginning, I'm studying interoception also using this discrete parareceptor firing. And as Sarah Garfinkel and Critchley and Seth found a few years ago, when you present fearful faces at systole versus diastole, it appears that they support the processing of fearful emotions, and that has given rise to the idea that interoceptive signals support fear. However, I really wanted to study this in a slightly different way and just understand if cardiac signals can support only fear or all emotions. So we published this last year, where I just took an emotional visual search task that had been developed by Lauren Umenma quite some time ago, and people had to detect an emotional face embedded with neutral distractors, and those faces were presented for a short period of time, and they were paired with systolic or diastolic cardiac phase. And then participants had to indicate where the emotion was presented and what type of emotion they found, and that they had been presented. And interestingly, we found that cardiac signals can support not only the processing of fearful faces, but also the processing of disgust and happy faces. And it looks like what it looks from here is that cardiac signals not only support fear, but really different emotions. And probably it depends on what kind of task you are using to study emotional processing. If you are using the emotional visual search task that we know shows a bias for happy faces, then people end up, or cardiac signals end up supporting this primacy effect of happy faces. So that's interesting. I'm running some replication studies on this, just changing some bits, adding metacognition to the whole idea. But this is interesting on its own, but I really wanted to do something with it. So we know that alcohol affects interoception. See the previous slides where we show these changes in alcohol cue reactivity and so on through interoceptive pathways. But we also know that alcohol changes emotional processing. It increases the perception of happy faces, and it dampens the processing of negative emotions. And that is one of the reasons why people end up drinking too much, just to modulate the way they perceive emotions in others. So what I wanted to test with this experiment is trying to see if people drink to mute their bodies and change the way they perceive emotions. So we are currently testing for this register report on drug and alcohol dependence. I actually just ran the 3rd session out of 80, so still a bit far from getting done. But basically, we'll just have this emotional visual search task, and people will come to the lab. We'll administer them alcohol, and we hope we will find changes in emotional processing as a function of how much systolic signals can amplify different emotions. And we are, of course, going to measure changes in ECG and heart rate variability pre and post alcohol administration to understand the physiological basis for any of these effects. So that's cool. That's interesting. But I'm working on a literature review about all the research that's been done on cardiac cycle and cognitive processing, and so far it's only very basic research, as in it hasn't really been applied to any problem outside of just trying to show effects of cardiac cycle on cognitive or emotional processing. So again, we know that binge drinkers have issues with emotional processing. They show emotional recognition deficits even as young as 15-year-old, or people with an alcohol use disorder or different addictive problems. They show differences in emotional processing. So right now I'm starting a collaboration with Pierre Morage, where we are going to administer this emotional visual search task to high binge drinkers and see whether the possible emotional deficits they are showing are due to an inability of interoceptive signals to support the processing of emotions correctly in these people. Maybe there is a disconnect of body-brain signals, and that is why these people are showing the problems in emotional recognition. Again, also with autism, there is more and more evidence showing that there is interoceptive deficits in this population. And of course, we all know there is emotional recognition deficits. So I'm running a small project at Rutgers as well, where we are administering this emotional visual search task with ASD participants to see again if emotional deficits are supported by interoceptive deficits. So that's interesting. But I'm also using this whole idea of presented stimuli with cardiac cycle to study addictive processes. So as I said at the beginning, there is an overlap in brain regions related to decision making and reward processing with interoceptive, with regions that support interoceptive signaling. And that has been going around for a while, but most of the research, if not all, is just based on neuroimaging studies. And I remember reading this quote during my PhD that the interoceptive representations of alcohol stimuli have the capacity to hijack the cognitive resources necessary for exerting inhibitory control to resist drug use. And I was puzzled by that. And really, I think it's very interesting to test that behaviorally. So that will also allow us to understand or to start filling in the blanks for this model about how more discrete cardiac signals or intermediate levels of interoception can support alcohol attentional biases. So what we are doing through a register report, it's actually under review, and a register report I'm written with Charlotte Ray. We are taking the emotional visual search task and we are replacing the emotional phases with alcohol cues. So participants will come to the lab and they will have to detect alcohol or neutral cues embedded in the emotional visual search task. And of course, stimuli will be presented at systole or diastole. And the idea is that when we present alcohol cues at systole, they will be more easily detected than at diastole compared to neutral cues. And what this will give us is the ability to understand if interoceptive signals can support reward processing in this particular population. So that sort of goes like one way, like interoceptive representation of alcohol cues or interoceptive signals can support reward processing, and we can test that behaviorally. But I am actually way more interested on the whole idea that these interoceptive signals can hijack cognitive resources necessary for exerting inhibitory control. So Charlotte published last year, a couple of years ago, a modified version of the stop signal task where go signals are presented at systole or diastole, or on the other hand, where stop signals in the stop signals task are presented at systole versus diastole. And what they found is that when you present stop signals at systole, people are better able to inhibit responses. Now, it turns out that you can also take a stop signal task, embed alcohol stimuli in it, and just have an alcohol stop signal task. And that's something that's been done quite a lot. So my idea with this project, as I said, it's under review. Hopefully we'll get it through. My idea with this project is, first of all, to embed alcohol cues as go signals that are presented at systole or diastole. And what we expect is that when alcohol cues are presented at systole, there will be a higher impairment in inhibitory control compared to neutral cues. And on the other hand, we will ask participants to come to another session where they will complete again this stop signal task with alcohol cues, but on half of the trials, the stop signal, on half of the stop trials, the stop signal will appear at systole or diastole. And what we expect here is that stop signals will facilitate inhibitory or response inhibition in the presence of alcohol cues compared to diastole. And what this can help us understand is if we can maybe use interoceptive signals to facilitate response inhibition in the future. So that's what's going on right now. I think it's pretty interesting. Hopefully we will find some cool results. So any questions before we move to another topic? Okay. What about aversive alcohol effects? So far, all the research I've been doing relates to positive reinforcement, and especially to alcohol attentional biases. So people trying to go and look for alcohol stimuli, like positive alcohol effects. However, there is much more in the picture. First of all, we need to look at negative reinforcement, which we haven't started yet, but something I want to get into. But we can also look at negative alcohol effects within a positive reinforcement perspective. And the idea for this project comes from this finding I was talking about before, that changes in interoception after alcohol administration may constitute an interoceptive experience that shapes negative expectancies. Now, anyone who's drunk alcohol knows that one of the worst negative effects of alcohol is hangover. So I just got a grant to study this. So we know that when you feel hangover, you feel pain in your belly, you feel your heart racing. At least I do. It's horrible. We know that hangovers have actually effects in cardiovascular functioning. You are thirsty, and all that to me is just an interoceptive process, really. And moreover, hangovers are also associated with negative emotionality and anxiety. So to me, I think there is a very large interoceptive component to hangovers. So that's what we are preparing a study right now where we will recruit 170 people online, and we will get binge drinkers who experience hangovers, who experience blackouts, so they drink quite a lot, and we'll just make a baseline measure, everything online, everything through the smartphone. We will do a baseline measure using the phase adjustment task to measure interoception that was developed by Jeff Bird, and a bunch of measures of alcohol history of alcohol use, mood, etc. We'll do that at baseline, and then on 3 different occasions when we know they've drunk the night before, we will ask them to do it again. And what we want to see is how, depending on hangover severity, we observe changes in interoceptive accuracy in the phase adjustment task. So basically, I don't really have time to go through the detailed expected results, but basically, if we consider that hangovers can be a very negative effect of alcohol, we would expect that greater changes in interoception will correlate with hangover severity, and also people usually will engage in less extreme alcohol drinking because their hangovers are so bad, and that's what one normally would expect. Except some people probably will keep drinking despite very negative effects on interoception. And that goes just to the basis of what an addictive process is. It's just persevering in the behavior despite the negative consequences. So we expect to have a bunch of people in here who will show very big changes in interoception during hangover and still persevere drinking, getting blackouts, getting strong hangovers despite all those negative effects. And also something we are testing. I have 2 computer science undergrads working on it. So I really like this work by Lauren Umenma, where they were able to map different emotions in the body by asking people to indicate where they feel changes in their body with different emotions. And I think we can use that to study the interoceptive components of hangover. So we are working on taking the task they developed and allowing people to do it from their smartphone. So when they are hungover at home, lying in bed, they are able to indicate where in their bodies they are feeling the effects of hangover. And this is just, we are just testing whether this will work. I'm also running this actually now that I'm doing alcohol administration. we're also looking at the effects of alcohol on bodily sensations in this way. So yeah, that is some of the stuff we are working on. And just finally, to move on to a slightly different topic, I'm just going to be talking really about more physiological stuff right now. So our lab has been working for many, many years on resonance breathing. Some people call it biofeedback breathing, biofeedback resonance breathing. There's different ways of looking into it. But basically, what we do is we ask people to breathe 10 times per minute. And what this does is this very specific frequency is important because it generates a coupling of phase in oscillations between breathing, heart rate, blood pressure, all using the resonance characteristics of the cardiovascular system. And what that does, it increases body-brain connectivity and baroreflex functioning. And this resonance breathing is important because it generates healthful changes. It strengthens the adaptive range and flexibility of the cardiovascular system in the face of physiological and cognitive challenges. And we know that it has huge clinical benefits. In our lab, we have been able to show that resonance breathing can decrease craving in people with an alcohol use disorder. And it also improves mood and emotional processing in people with depression and anxiety. So we don't really know how it works. We know how resonance breathing works. Basically, our hearts have this natural frequency at which they oscillate, which follows the delay in the baroreflex. That is just like 5 seconds up, 5 seconds down. So that's 0.1 hertz. So when you are breathing at 6 times per minute, you're just pushing this oscillation at the right spot and just amplifying it, really, and just pushing your cardiovascular system to oscillate further in that direction. Now, the idea for this, I had it when I was in the lab, and I was coding a pacer on the screen, and I started following it, and all of a sudden, I just could feel my heart much, much more. And that gave me the idea that if resonance breathing can increase baroreflex sensitivity and also 0.1 hertz heart rate variability, because it just occurs at this very resonant frequency of 0.1 hertz, and both these indexes reflect afferent cardiac signaling, well, then maybe we can modulate interoceptive awareness using resonance breathing. So we did that through a register report that just got published, and it was an absolute ordeal because I had to test through COVID times, and it was very difficult. So I'm very happy this came out through. So basically, we asked people to come to the lab, measure baseline interoception, then physiological changes, physiology at baseline, and then we made people breathe 6 times per minute or a controlled breathing task, and then again more interoception after the breathing tasks, and just putting some more breathing in the middle just to make sure that we're really amplifying the cardiovascular system. And of course, we also wanted to study just the basic physiological correlates of interoceptive awareness. And what we found was very, very puzzling at the beginning. We found the opposite of what we were expecting. We found a negative correlation between heart rate variability and interoceptive accuracy, and we were expecting the complete opposite. So that was really weird. But we'll see in a second how we can understand this. However, very interestingly, we found that the more the cardiovascular system aligns with the resonant breathing effects, the more 0.1 hertz heart rate variability increases after resonance breathing, and the more baroreflex sensitivity increases after resonance breathing, the better people are at feeling their own heart. And that was great. That was a positive correlation between interoceptive accuracy and changes in physiology, as we plan in the register report. So how does one integrate both findings, the negative relationship between heart rate variability at baseline and interoceptive abilities, and on the other hand, the fact that if we increase 0.1 hertz and baroreflex sensitivity, we get more interoceptive accuracy. And this is where ideas of predictive coding and interoceptive inference come in. And actually, there is more and more work. Actually, Charlotte Ray did it in her paper. And also Andrew Corcoran has been finding that heart rate variability decreases, or changes in, or less heart rate variability facilitates interoceptive processes or cognitive processing. And the idea is that if we are constantly striving to reduce prediction errors, and we have shown that, of course, with exteroceptive processing and proprioceptive predictions, if we are constantly striving to reduce prediction errors to facilitate perception of external or internal stimuli, then it will make sense that less variability in the signal, less heart rate variability, will generate less prediction errors to increase this interoceptive accuracy. And it is actually way easier to understand this point when we look at what our heart does during resonance breathing. So this is RR interval. So this is heart rate variability at baseline. And you can see that this is just basically noise. There is some cyclical variability here. I think this is probably representing the bump here at 0.28 Hertz. So we can see some cyclical variability, but it is also quite random, you know? So imagine I am a brain trying to make a prediction as to when the next heartbeat will occur. I'm gonna have a very hard time, right? And this is what happens when people start breathing at 0.1 Hertz. You can see this is respiration going at 10 times per minute. And this is the variation in RR intervals. And what we see is that changes in RR intervals just fall within this very predictable oscillatory pattern at 0.1 Hertz. And your heart just starts cycling following your respiration. So if I was a brain trying to establish a prediction about when the next heartbeat is going to occur, just to build a successful model, obviously I'm going to be much more successful in this case. So this is why we think that less variability in here is going to improve interoceptive accuracy at baseline, whereas more variability within 0.1 Hertz is going to increase interoceptive accuracy after breathing. And that just raised a lot of questions for us, because we don't really know why resonance breathing works as a treatment, as an adjunct treatment tool. And we don't really know either why heart rate variability is lower in mental health disorders. And people always say, you know, like heart rate variability is lower when you have a mental health disorder or whatever, just because of stress. But actually, again, Corcoran and others have this idea that when you are facing uncertainty and stress, our brain just downregulates heart rate variability to reduce these interoceptive prediction errors and facilitate cognitive and emotional performance under stress. So the reason why heart rate resonance breathing might work in clinical populations is because it improves interoceptive prediction errors, not because it makes heart rate variability larger, really, but because it forces the heart to oscillate at a very predictable pattern, and in this way improves emotional and cognitive processing. So maybe resonance breathing was always partially about reducing interoceptive prediction errors. Again, need to replicate these findings, but I think it's quite interesting. So anyway, almost done. That's right on time. So just like I've been talking about a lot of stuff, and probably you're wondering like, what does this have to do with one another, right? But actually, I think there is a bigger picture here. Today I've been able to talk about how we've been able to show that cardiac signals support emotional processing, and how there is an interoceptive basis for alcohol-related behaviors, and how we can modulate these interoceptive processes via resonance breathing. What I'm testing right now is trying to see if there is a basis for different clinical disorders through this emotional processing and interoceptive signaling. We are also studying whether cardiac signals can support inhibitory control and attentional biases in alcohol in binge drinkers. So the next step really is to try and see whether we can improve the support of cardiac signaling in emotion and in inhibitory control via resonance breathing. Actually, this week I'm starting to test this, whether I can improve the ability of interoceptive signals to support emotional processing using resonance breathing. At some point, we'll also have to do the whole thing with the cardiac stop signal task and resonance breathing. And if that works, then, well, maybe we can start getting into the serious stuff. And we can start looking at whether resonance breathing can improve the link between interoception and emotion in clinical populations, or maybe we can improve inhibitory control in alcohol use disorders using resonance breathing by tweaking interoceptive pathways. And with that, thank you very much to everyone at Sussex who's helped me, and to my team at Rutgers, and of course my bosses for letting me do all this stuff. Thank you.
Thank you very much, Mateo. Does anybody have any questions? Jess?
Yeah, hi. That was a really lovely talk. I was just wondering, when you were presenting about the stuff that you were trying to do, or you did about hangovers, how did you manage to capture interoceptive awareness on a mobile telephone?
Good question. So we are using the task that Jeff Bird has developed. Jeff Bird is at Cambridge, if I'm correct. He has developed a task with David Plains where they are, it only works on iPhone, but basically you download an app, you put your finger on the camera.
Oh, finger on the camera.
Yeah. And then there is a rotary, like people are presented with a feedback tone, and they have to synchronize the feedback tone with their cardiac signals. And it just gives an index of interoceptive accuracy.
I see. And is this commercially available, or through collaboration?
You just have to ask him. He doesn't ask for authorship or anything. He just wants people to use it. It's really helpful. Yeah.
Does it measure the cardiac signal through the camera, through the skin?
Yeah.
Very cool.
Yeah. Really cool. We are very excited.
Anybody else? Dora.
Hi, Mateo. Very, very impressive. So many aspects that you mentioned today, and really, really gave a lot of maybe ideas and thoughts. Just listening to your alcohol effects and how you modulated or you found about the differences in interoceptive accuracy and awareness. I was just wondering, you haven't looked at the same time in any other traits that maybe exist, like, emotional dysregulation. I was thinking actually about what we have been doing with alexithymia, but not alexithymia, something maybe about empathy or some other type of trait, emotional impact that maybe you find. I noticed that you have looked in as a trait only in family history of alcoholism, but I was wondering whether either you have measurements that you have looked and you didn't find anything, or whether you plan to look. You mentioned that you will look in binge drinking and emotions, and there are a lot of studies about alexithymia, empathy, and whether you are planning in your future studies to enter some of these questions. This is actually one of the things that I thought that somehow was missing. Also about your negative expectancies, I was thinking whether these people were more anxious or more, more with negative baseline thoughts or emotions, and how that may contribute in some of your alcohol effects. So do you have any thoughts about that or any plans about that?
Yeah, thank you, Dora. Yes, about expectancies, absolutely. We need to replicate that. It's not clear. And actually for this new experiment I'm doing, the one we just started last week, we are actually asking them to complete the alcohol expectancies 1 or 2 weeks before the experiment. Like they have to do it at home, completely detached from the experiment. So we can actually just separate the maybe anxious component of, am I about to get alcohol in the lab? Or, so we can really try to separate both things. People are doing it at home, and we will see if we replicate. Because I think it's a cool finding, but absolutely needs replication. Yes.
And you could maybe measure the mood or something, baseline mood before they enter into the session. But anyway, yeah. Yeah.
We have panas. Yeah, we have panas at baseline. I mean, we have panas throughout the entire procedure. Yes. Yeah. And about the alexithymia thing, I did not measure it on the alcohol one, the one I'm doing right now, and it's probably too late. But that is a good point. I will just have to do it again. Yeah, I didn't measure. That is a good point. I've measured alexithymia in, I think, in the very first emotional visual search task, like the very first time I did it, I think I measured alexithymia and didn't find anything.
Okay.
I think. Yeah. No, absolutely. Again, there's just a lot of, like, I'm already doing a lot of questionnaires, there is a lot of stuff. So there is a limit. But yeah, no, you're absolutely right, Dora. Yeah.
Because especially, actually, I was thinking also as a practical more comment about your hangover people, because they will be in such a negative mood and they are so much concentrated in their bodies that one really needs to tease apart how much this, emotional aspect during this hangover may contribute or may change some of your findings. It is good if you were able to identify it. That's anyway. So
I mean, when they are hungover, we will be measuring mood and how anxious they feel and the negatives. Yeah. So that's the whole idea. The whole idea is those who show more changes in interoception, more disruption, and that's the thing. I say changes because I don't know in which direction it's gonna go. I, for example, will feel my heart so much more. So I purposefully don't say decreases or increases. I'm probably gonna go for absolute change because I don't know, how it's gonna play out. It's a very exploratory experiment, really. So yes, that's the whole idea, that the more there is an interoceptive disruption, the worse their mood is gonna be. Absolutely. So we are measuring that via PANAS, STAI, hangover, acute hangover effects. Yeah, that's the whole thing. Yeah, absolutely. Thank you.
Can I come in, Chris?
Yeah, please do.
Hi, Mateo. Wow, it was just so great to see the whole body of work, and I really learned a lot, and the resonance breathing in particular I found really interesting. So I have a question about individual differences, because you showed loads of work, across all your different studies that taps into different levels of lower order, such as the 0.1 Hertz frequency channel through to higher order. And this seems to interact in different ways with how people feel and think and behave. So it's really got me thinking about different groups of people, like social drinkers, binge drinkers, alcohol use disorder, people who don't drink at all. And what is it that leads to different motivation for drinking in these different groups, and maybe different inhibitory capacity as well? Do you have, like you showed the lovely model diagrams, do you have like an overall model of this is what drives alcohol use disorder, this is what drives binge drinking, in terms of those different interoceptive channels?
Yeah, thank you, Charlotte. Yeah, I don't know. We just need a few more years. You're absolutely right. Like, I don't know what shapes each of these things. Like, there's only, like, literally, there's only 3 papers. You know what I mean? We need to research it much more. I will say, like, in line with what you're saying, there is a whole aspect, and I say it, like, we haven't looked at negative reinforcement, and I think that's the key. We need, so far we're just looking at alcohol attentional biases, etc. Like, we need to start looking at interoceptive deficits in alcohol use disorder, like clinical populations, and seeing how their bodies are changing. So for now, the thing we are working on with my PhD student, I didn't talk about it because it's very early stages still, but we are gonna take cannabis users, like people who smoke a lot, people who drink a lot, and people who crossfade, who do both. Gonna bring them to the lab, and we are gonna stress them out and see, like, using an arithmetic task, and see if they show differences in how their bodies change, especially the 0.1 Hertz. So see whether each of those groups are motivated to drink or to smoke weed, or like just different consumption patterns depending on how their bodies are affected by stress, because that is just a way of, stress is a big risk factor really in any addiction. So that is one way we are starting to look at it, but we need a lot more work.
Yeah, no, that's really exciting. But yeah, it sounds like the 0.1 Hertz channel is a good place to start for that.
There's a question in the chat about this from Bex Atkinson. Really impressive body of work, Mateo. Maybe I missed this, but what is the mechanistic relevance of 0.1 Hertz? Her camera's not working, so she can't ask by chat.
Hi, Bex. Yeah, no, that's a very good question. So I explained very quickly, but I didn't explain enough. So 0.1 Hertz. So your heart oscillates at different frequencies, right? And mostly when you're at rest, it oscillates at high frequency and also low frequency. But there is a delay of 5 seconds between when your baroreceptors are signaling to the brain that there is a change in blood pressure, and then when your brain is signaling back to the heart to change heart rate as a function of this change in blood pressure. And that takes 10 seconds, give or take. So 10 seconds is 0.1 Hertz. So that is the reason why we consider this 0.1 Hertz index as just a proxy for baroreceptor sensitivity. So when we do not have baroreceptor sensitivity measures, which happens quite often because it's very, very expensive equipment, then we just use 0.1 Hertz as the relevance of 0.1 Hertz it's just a proxy for baroreceptor sensitivity. The good news is that I just got a bit-to-bit blood pressure machine, so I can now have baroreceptor sensitivity on all my experiments. So that is very good. But we will have, again, the 0.1 Hertz, and the idea that it reflects, the idea with 0.1 Hertz is actually that it reflects afferent cardiac signaling. Like, it's just a way of quantifying how much information goes from the heart to the brain. Now, that was postulated in the 80s, and since then, we really need to look into it. And that's something that, again, it's like endless amount of work. But yes, absolutely. That's a good question.
Thank you very much. I think maybe we've got time for one more question. Does anybody else have a question? Zoltan.
I was interested in the resonance breathing you mentioned. I was wondering if the name is a bit grand. It sort of implies a theory that you said really wasn't there. I mean, if resonance is really the issue, then if people breathe that 5 or 7 times a minute, you'd no longer be in resonance, but 12 times a minute would be resonance, other frequencies staying the same, which I know they wouldn't. I mean, so would just slow breathing be a better name? And what was your control group, and what did it actually control for?
Okay, thank you, Zoltan. So there is something special about this. If you make people breathe at 0.15, you don't get this massive spike in heart rate variability. You make people breathe slightly slower, you don't get this massive spike. There is something special. So that's why we don't.
So you do get people breathe at 5 and 10 times, at 5 and 7 times either side of it. And then
I haven't done it. I haven't done it. Others have done it. Others have done it. Yeah. This is a well established. I mean, the guy who pretty much developed it, Evgeny Vashilo, he was a professor in our lab. He passed away last year. He's the guy who developed this. So he has done all the testing, and this massive spike in heart rate variability, which we can see here in time domain, just all these RR changes really following respiration. If you make people breathe at other frequencies, they do happen, but the change is not that massive. Like this is on scale. These two axes, this one and this one, are on scale. So it's just to show you that the effect is very, very strong. So if you make people breathe at different frequencies, you do not get this effect. Another interesting fact about resonance breathing, and we can barely see it here, but as a resonant effect, it also has harmonics, like when you play an instrument and just the note decomposes in multiple harmonics. So when you make people breathe at resonance breathing, you see small bumps in heart rate variability at these other frequencies dissipating little by little, just as if you played a musical note. So you can see here the little bump. So it just generating a lot of harmonics as well. And that is just showing again that there is something special about this. As what I used as a control for this particular experiment, it was actually within, so they came for 2 sessions. I made people breathe at variable breathing because I was concerned about what you're mentioning. I was concerned that we will generate some sort of resonance effect if I made people breathe all the time at the same frequency. So what I did is people were breathing from, I don't remember what it was, from like 10, I think it was, yeah, from 10 to 15 times a minute, just cycling. Like every breath will be slower than the previous one up until they reach 10 breaths per minute, and then again faster until they reach 15 times a minute, and just oscillating throughout the 5 minutes of the measurement, just to make sure that I'm asking people to breathe at a certain frequency and pacing their breathing. So it's a control task, but I am not engaging in any continuous possible resonant effects at other frequencies. Because yeah, there might be some resonant effects, as you mentioned, although definitely not as big as this. Yeah, make sense?
Yeah, thanks.
Thank you.
Fascinating stuff. Thank you very much, Mateo, and thank you everybody else for coming along. And it's great to hear about what you've been doing. So hopefully you can come in person in a couple of years and update us on all of these ongoing studies.
Yeah, that would be great. Okay, thank you, everyone. Really happy.
Bye, Mateo. Bye.
Bye.
See you again.